Good morning, students. So yesterday, our last class, we discussed the pistil, the gynoecium part. The gynoecium consists of pistil, megasporangium, or ovule, and pollen gland. Sorry, embryo sac. Embryo sac. Here, the pistil, it consists of three important parts: stigma, style, ovary. The stigma is pollen receptive, where the pollen grain is germinate. The style is a long, slender, a tubular structure where the pollen tube is passes towards the ovary. The ovary, a bulging portion of a pistil, where the location of ovule or ovules. The ovules arranged in different manner. That is what we have discussed, the types of placentation. So that is after pistil, we discussed the megasporangium or ovule. Here, megasporangium or megasporangium, it is also called a female gametophyte. Female gametophyte. Because the megasporangium, it consists embryo sac. The embryo sac consists of a female gamete called egg. As it is a female gametophyte, also there is megasporangium is also called ovule. That ovule which developed as seed in future development. As we study the detailed structure of ovule or megasporangium. Here, each and every megasporangium which consists certain important parts. So those are a funicle. Funicle is a stalk of ovule. That is, funicle is stalk of ovule. So these stalk, it is originated from the placenta. Placenta is a white colored tissue which is present in different positions in the ovary. On that placenta, there is organization, there is origination of these ovule or megasporangium, which consists of a short stalk. A short stalk, that is what we are called as funicle. Funicle is stalk of ovule which originated from the placenta. As even in the funicle, there is presence of a vasculature. There is a presence of vasculature. There is a vascular tissue. It is nothing but there is supply of water and food towards the ovule. So that is what we are called raphe. The raphe which runs from that is funicle. These funicle which attach to a body of ovule. The funicle attached to body of ovule, the junction between that is funicle and body of ovule, it is called as hilum. That is junction between body of ovule and funicle. The junction between funicle and body of ovule, it is called as hilum. It is nothing but hilum is a point. Hilum is a point where the funicle, where the stalk attached to ovule. As the body of ovule, which covered with two integuments. How many integuments? That is two integuments. These two integuments are outer integument and inner integument. The body of ovule covered with two integuments, that is outer integument and inner integument. Inner integument. So commonly called as integuments. Here, the ovule which covered with either single or two integuments. It is nothing but if the ovule which consists or which covered with a single integument, it is called as unitegument ovule. Unitegument ovule. So major monocots which consist unitegument ovule. Unit is nothing but single. Tegma or tegment, it is nothing but covering layer. That is unitegument ovule, it is nothing but the ovule which consists single tegma or single covering layer. So that is unitegument ovule, it is nothing but ovule consists only one integument. If maybe like what we have the diagram, it consists two integuments. These are outer integuments, these are inner integuments. As these two integuments present in the ovule, such ovules called as bitegmic ovules. Bitegmic ovules. So most of even dicots and certain monocots which consist, the ovule consists of two integuments. The two integuments which are present in the ovule, which are called as bitegmic ovules. So these classification is based on presence of the number of integuments. Here, that is the classification, that is two types, unitegument ovules, bitegmic ovules, that is based on these number of integuments. Sometimes if the ovule is not consist of any covering layer, there is no covering layer, that is what we are called as ategmic ovule, that is nothing but absence of tegma, absence of covering layer. So those ategmic ovules are nothing but the ovule which does not possess any covering layer. As here, if in the bitegmic ovules, what the covering layers which protect the entire embryos are present inside the nucleus. Sometimes below the integuments there is presence of nucleus, sometimes the nucleus is absent. At the time of nucleus is absent, the inner integument it is also possess a larger cells which nourish the embryo sac or which nourish the female gametophyte. As well, sometimes the inner integument it acts as nutritive tissue. Not in all conditions or all times, but exceptional conditions where the nucleus is absent in the ovule. So, ಯಾವ ಓವ್ಯೂಲಲ್ಲಿ ನ್ಯೂಸಿಲಸ್ ಡಿಸಪಿಯರ್ ಆಗಿರುತ್ತೋ ಅಂಥವರಲ್ಲಿ ಅಂಥ ಓವ್ಯೂಲ್ಸ್ ಅಲ್ಲಿ only the inner integument, not outer. The inner integument it acts as a nourishment tissue which possesses larger cells and which store the nutrients, which store the reserved food materials. So that is the part of integuments. These outer and inner integument in future which developed as seed food. A seed, every seed is covered with a seed food. For example, why you take a groundnut seed? Even the groundnut seed, the seed food is differentiated. The color is differentiated from outer side and inner side. So easily observe that seed food in tamarind seed. In tamarind seed, outer little bit dark color. So the same seed food that is inner, it is cream color. That is color is differentiated. That color is differentiated due to that ovule or the tamarind seed or groundnut seed, it is developed or it consists the two integuments, outer integument and inner integument. These two integuments which take part in formation of seed coat, the seed coat is also differentiated, that is two layers, outer stigma and inner testa, that is testa and stigma. These two layers which formed by the outer integument and inner integument. So the testa and stigma which is easily differentiated in the, that is the seed coat of the tamarind seed. So that's why what those the two integuments which are formed by the two integuments which cover the, that is the ovule as it differentiates, as it developed, that is seed coat in future development of that ovule. That is ovule developed as seed, what the seed is, that is seed consists of different parts. So as inner to these seed coat, there is presence of nucleus. Nucleus is a nutritive tissue. A nutritive tissue. So these nutritive tissue which consist of larger cells. The cells are larger, dense cytoplasm, prominent nucleus, like those nourishment tissue which present inside the inner integument or inside the integuments. So as the students, you remember, if any nutritive tissue may be either multinucleate or even uninucleate. If it is uninucleate, the nucleus is larger in size and the reserved food materials are stored, hence which consists of dense cytoplasm or the cell size is larger. So hence the nucleus is a nutritive tissue which nourish the developmental stage of embryo sac. which nourish the embryo sac, which supply the food for development of embryo sac. Because the embryo sac development, it takes in the nuclear tissue. Nuclear tissue, we discussed that how the embryo sac is developed. So at the time of development, the cell of one of the cell, which is the larger sized cell, which is archisporium cell, that cell divided peritoneally, it leads to formation of two cells, that is outer parietal cell, inner sporogenous cell. That cell is take part in, it leads to formation of a complete embryo sac. How the embryo sac is developed, we can discuss it. So at the time of development, they required energy, they required food. That food is supplied by these nutritive tissue. That is, nucleus is a nourishment tissue or nutritive tissue which is present below the integumentals. So it is also an important tissue present inside the ovule. So because in the development of these ovule into seed, that is, it requires food nutrients. So that is presence of nucleus. So after these nourishment tissue, a complete development of seed, that nucleus is either completely consumed, either maybe certain amount of nucleus is remained. If after complete development of, that is, the ovule is developed into seed, even the seed is also consists certain amount of that is nutritive tissue, that is nucleus. Such seed is called as such a perisperm. The remnant of nucleus, remaining amount of nucleus present in the seed after development of complete seed, that is what we are called as perisperm. As maybe sometimes every ovule which consists a single embryo sac. So one of the cells of nucleus cell is only take part in development of these embryo sac. The embryo sac, it is developed by one of the cells of nucleus. Sometimes a seed consists more than one embryo sac, as we discussed that is polyembryonic, that is in citrus plants, or that is lemon, orange, which consists more than one embryo which is present in the seed. So there is nothing but there is presence of number of embryo sacs, or number of that is the embryo sacs in each ovule or each seed. A single seed contains more than one embryo, that is what the phenomenon called polyembryony, in which it is take part by, that is malfunctioning. That is nothing but maybe of the certain number of cells which is developed as embryo sac. That is formation of more eggs in the each embryo sac also, or maybe of number of embryo sacs formed. That is, those egg cells present in the number of embryo sac maybe get fertilized. So during which there is formation of number of embryos, more embryos are formed. So that is due to certain cells of integuments also, it can take part into formation of embryo sac. Number of cells of nucleus is also take part in formation of that is embryo sac as it leads. So here, just below the integuments there is presence of nucleus. Inside the nucleus there is presence of embryo sac. So it is the embryo sac where the embryo is developed. So as inside the embryo sac, there is presence of 7 cells. 7 cells. So we need not write in the same diagram. So it is enough. Just I show the cells of embryo sac. It consists of 3 egg apparatus, 1 central cell or diploid polar nuclei, and the 3 cells which are present in the callosal region, those are called as antipodals. So we discussed this one also, that is in the next. So it is the embryo sac. As that is the entire ovule which consists of 2 ends, that is chalaza. It is nothing but broad base of ovule is called as chalaza. Broad base is called as chalaza, and the body of ovule which covered with the 2 integuments by leaving a small space. This small space is called as micropyle. So the micropyle is nothing but a place or a point where absence of integuments. The body of ovule covered with integuments, where the integuments are absent, which are not covered completely, where there is a space. That space what we are called as micropyle. So what we say is that is the ovule which consists of 2 ends. Those 2 ends are the ending towards the chalaza is called as chalazal end. The end towards the micropyle, it is the micropylar end. So here, micropyle is the space or a point or a place where the absence of integuments. These micropyle, it is formed by either both integuments, either single integument, that is either inner integument or either outer integument. So that is nothing but, for example, so like here, only the outer integument only it leads to formation of a micropyle. That is a pore formed by outer integument. This pore what we are called as exostome. So either maybe that micropyle is formed by that is see here, the most of the micropyle it leads to formation of by only inner integument. It is outer integument, it is inner integument. It is outer, it is inner. Here the micropyle formed by outer integument. Here that micropyle formed by inner integument. So this type of micropyle is called as exostome. It it is yellow stone. It is nothing but based on the micropyle. It is a pore. It is a pore. It helps to entry of polar tube during the fertilization. While the polar tube entered into the ovule, these micropyle is formed by either both integuments or any one of the integument. So that is either by outer integument or inner integument. If the micropyle formed by outer integument, it is called as exostome. That micropyle formed by inner integument, so that is what we are called as endostome. So here, the end, the two ends, that is chalazal end and micropylar end, those which facing towards the chalaza or base of the body of ovule, or towards the polar region or towards the pore region, that is micropylar end. Students, it is the megasporangium, or it is plural megasporangia, or it is ovule. Is a singular, ovules plural. So the structure of entire megasporangium. So once again we discussed the entire structure of that megasporangium consists funicle. Funicle is a stalk of ovule where it gets attached to the, that is, placenta. So these funicle which get attached to the body of ovule, it is what we are called as a point. A point attached to the funicle attached to body, it is called as phylum. The body of ovule which covered with the two integuments, that is outer integument and inner integument, which developed as seed coat in future development. So the body covered by these two integuments, by leaving a small space or place, or there is a pore formed, what it is called as micropyle. It is a pore. It is a pore. And it is also a space or place where the absence of that is the integuments. Inside the integuments, presence of nucellus. The nucellus is a nutritive tissue which supplies the food for development of embryo sac, or at the time of developmental stage. Inside the embryo sac, there is presence of 7 cells, that is 1 egg, 2 synergids, 1 central cell, 3 antipodals. As it consists of 2 ends, that is micropylar end and chalazal end. Chalaza is the broad base of the ovule. So as it is one of a type of anatropous ovule. Anatropous ovule. So we identified the different types of ovules. The types of ovules based on position of funicle, and micropyle. Based on position of funicle, that is the stalk of ovule, and the presence of pore or micropyle, there is different types of ovules. So here, so these the types of ovules. There are anatropous ovule, hemi-anatropous ovule, ovule, orthotropous ovule, orthotropous ovule, campylotropous ovule, campylotropous ovule, that is amphitropous ovule, the last one is succinate, ovule. So these are the different types of ovules. Ovules. See here, based on important, that is the position of funicle and micropyle. Position of micropyle and funicle, there is the different types of ovules. The first one, that is anatropous ovule. As you see here, that is here, it is the funicle, it is the micropyle. So in anatropous ovule, the funicle and micropyle, it is little bit near, not more near. So as the body of ovule, it is little bit curvature, it is seen from orthotropous to anatropous. So as considered in orthotropous ovule, in which chalaza, micropyle and funicle. So these three all are present in the same line. See here, it is the micropyle, it is the chalaza, it is the funicle, which are present in the same line. While compared to orthotropous, in anatropous ovule, the micropyle, which is present near to funicle, while we compare to orthotropous to anatropous. In anatropous ovule, the micropyle present near to funicle, As in orthotropous, all these three which are present in the same line, so it leads to formation of 180-degree angle. 180-degree angle, where that is micropyle, chalaza, funicle. So as that is, we identified certain differences in the different types of ovules. So as in orthotropous ovule, all these 3 points, that is micropyle and funicle, it leads to formation of a straight line. So that is formation of 180-degree angle. In anatropous ovule, the micropyle is near to funicle, as we shown in the diagram also. So we write there in the inverted, we write here upright. So both are same. So this one and this one are same. That is, both are anatropous ovules. So it is orthotropous ovule. So after semi-anatropous ovule. In semi-anatropous ovule, see here the diagram, that is nothing but as widely compared to anatropous and hemianatropous. The funicle which lies perpendicular to axis of micropyle and chalaza. There is nothing but the micropyle and chalaza present in the same line. The funicle which lies perpendicular to these axes. That is nothing but the right angle is formed. That is 90-degree angle formed here. So these funicle, it is also right angle to intermediates, right angle to axis of embryo sac. So everyone, that is the funicle is present right angle to these. So where the position is little bit far from funicle to micropyle. So this type of ovule, it is called as hemianatropous ovule. Third one, orthotropous ovule. Already we discussed that those are all in which all the chalaza, micropyle and funicle which lies in the straight line. The next one is campylotropous ovule. The campylotropous ovule, here the curvature, it is nothing but, for example, it is the hemianatropous, as it is the anatropous. The body of curvature is little bit more. See here, the body of ovule, it is little bit curved nature. That is nothing but the micropyle is so nearer, that is very near to funicle, because these body of ovule, it is little bit more curved. So such a type of ovules what we are called as campylotropous or campylotropous ovules. So another one is amphitropous ovule. So just here you observed there is campylotropous ovule and amphitropous ovule. In amphitropous ovule, here also more nearer to, that is micropyle is very near to funicle, but the body of ovule, it is more curved, very curved. So that is nothing but the body of ovule, it is very curved or half-moon shape, or here the embryo sac is also horseshoe shape. That is nothing but here. So embryo sac is, at the time of body of ovule is curved, even the embryo sac also shows curve. The curve looks like a horseshoe shape. So because of more curvature present in the, that is amphitropous ovule. Amphi is nothing but both. Here there is presence of the two ends, it is little bit nearer, that is the ends of embryo sac. So as it shows curved nature, that is horseshoe shaped. So that is the main nature of ovule of that amphitropous ovule. So another one is the remaining, the last one, that is you see that is in the pumpkins or cucurbitaceae family, that is sarcinate ovule. Here the body, it looks like a, that is orthotropous ovule, but at the time of origination there is here funicle, it is a micropyle. These, the curvature of body of ovule, the curved nature of body of ovule, which turned one complete turn, as at the time of curved nature, so now it is cunical, it is C. That is nothing but a orthotropous ovule which curved like, that is 360-degree angle. 360-degree angle. One complete turn is turned by this the ovule of succinate. So that type of ovules which are called as the succinate type of ovules. Succinate type of ovules. So these are the different types of ovules. The different types of ovules, the classification of ovules. So based on position of cunicle and micropyle. The different types of anatropous ovules, hemianatropous ovules, orthotropous ovules, campylotropous ovules, amphitropous ovules, and succinatropous ovules. In anatropous ovules, the micropyle is near to cunicle, fully near. Hemianatropous ovule, the body of ovule is little bit curved. So means the turn, at the time of turning, the axis formed, the line formed from micropyle and chalaza, which right angle to present, that is cunicle. Cunicle present right angle to the integuments or line of embryo sac or the line of micropyle and chalaza. In orthotropous ovule, micropyle, chalaza and cunicle which rise in the straight line. Campylotropous ovule, here the body is curved nature, so the micropyle is nearer to cunicle. In amphitropous, the micropyle is very nearer because of that is the curve of body is more curved. Here in that amphitropous ovules, the horseshoe-shaped body or horseshoe-shaped embryo sac is identified. In succinate type of ovules in which the entire body of ovule which turned one complete turn. So the turn is little bit one complete round or 360-degree angle. So here also there is a micropyle, chalaza and cunicle which is present in the same line, but it is little bit variant. It is complete turn of cunicle. So this is the difference of different types of classification of ovules. So depend upon based on position of cunicle and micropyle. So next, a female gametophyte, where the female gamete is present in the embryo sac. An embryo sac, it is formed by one of the cells of nucellus. That nucellar cell, which is mustard shaped, larger in size. That nucellar cell present towards the micropylar area. The nucellar cells already which are larger size. So even though which cell which donate, which cell it leads to formation of that embryo sac, it is little bit more larger than the remaining cells of nucellus. As here the embryo sac, it leads to formation of either by single megaspore or 2 megaspores or 4 megaspores. So that is nothing but what the megasporogenesis. Megasporogenesis. The megasporogenesis is nothing but the process of formation of megaspores from megaspore mother cell. Megaspore mother cells or megaspore mother cell is called megasporogenesis. Megasporogenesis. So there is formation of female gamete. As the male gamete in the size very small, that is microsporogenesis. That microspores formed by microspore mother cell. As the megaspores, the process of formation of megaspores from megaspore mother cell are also it is MMC. MMC is nothing but megaspore mother cell. These megaspore mother cell is always that is diploid condition. But what the spores form, which is haploid. So here a megaspore structure of embryo sac as it leads to it consists seven cells as in the chalazal end it consists once again the three cells. In the center there is presence of a single cell. It is the chalazal end. It is a micropylar end. Every embryo sac consists of two ends. That is, as the ovule, the embryo sac present in the ovule. What the ovule it is has chalazal end and micropylar end. The chalazal end it consists three cells. Those three cells are called as antipodals. Or antipodal cells or antipodals. So here central cell or it is the polar nuclei as it is egg cell. These both are synergids or synergids. So combinedly called egg operators. As under synergids there is presence of filiform apparatus. In the embryo sac, towards the chalazary end, the three cells are located, which are called as antipodals or antipodal cells. The three cells present towards the micropylar end called the egg apparatus, in which one is egg cell, two are synergids. Centrally location of the central cell, it consists of two polar nuclei. So in the embryo sac, all the antipodals and egg apparatus are haploid nature. Only the central cell or polar nuclei, it is the diploid nature. It is 2n nature. It is n. It is n. So egg cell is also n. That is the haploid nature. So as this type of embryo sac, which is developed in the ovule. So how these embryo sac is developed inside the ovule, that is the developmental stages of embryo sac. Along with this structure, once again we discussed in next class. Thank you.